Hey everybody, this is the first of what should be 3 videos for Unit 2, Subunit 2's notes. If this is finding you on a snow day, I would like you to make sure that you're looking at the transcription and translation portions, which should be the first 2 videos. If this is finding you and you're one of my online learners, you're gonna have to watch all of them at some point. But I'm gonna upload them to everybody's classroom so you'll all have access to these. And the first video will be this directly into RNA transcription, which is the process of making RNA from DNA. The second video will be on translation, which is taking that RNA and turning it into a protein. And the third and final video will just be the wrap-up stuff about mutations and whatever's in the end of the notes. So we're going to go straight into the transcription portion of the video. So we're going to pause this and pick up over at the table. Okay, so to start for video 1, I've got some
Okay, go ahead, sir. Go ahead.
So for video 1, I've got some main ideas and some vocab terms here. I'm not necessarily going to stop and mention each of these things right now, but I thought you could kind of use this as a list. You could stop the video, write them down if you wanted to, but these are kind of the essential vocab terms from the first 5 pages. So I'm going to go a little bit more in detail than this with the packet here. So definition for transcription tells you what it is, talks about RNA a little bit, and then so this is going to be a probably 3-point thing on your quiz because there's 3 types of RNA called messenger, transfer, and ribosomal RNA, commonly abbreviated mRNA, tRNA, and rRNA using the first letter of each word. I would like you to know the shape of each one. Messenger RNA is a single uncoiled strand, so it would just be a little line. Think double helix, but only half, because RNA is single-stranded, not double-stranded. Transfer RNA, which forms kind of a little T shape. It's going to kind of look like this a little bit. So there's going to be some nitrogen base pairing in there. This isn't the best drawing I've ever made. And then these are going to be unpaired, because remember, it's still a single strand. And you can kind of see where maybe it makes a T shape if you think so. And then rRNA, ribosomal RNA, does not have a defined shape. So it's just kind of, a blob. My son makes artwork that kind of looks like that. Keeps going. Again, types of RNA, know those things. Tells you what a codon is. We've talked about codons in the past. It's a series of 3 mRNA nucleotides, and they're going to code for a specific amino acid. We've talked about amino acids in the past too. We'll get to that when we talk about translation a little bit more. And then I said to you guys last time that transcription was a lot like DNA replication, so that if you learned about replication, that this would be easier. Again, more vocab. Know what a promoter is. It's just a region of the DNA that tells transcription where to start. The enzyme doing that is called RNA polymerase. And then again, I'm not going to read the whole thing to you guys here. I'm just kind of hitting highlights. Remember, you have uracil with the RNA being used instead of thymine. So adenine is going to be pairing with uracil, not thymine, when we transcribe our RNA. Our RNA is going to be made in a 5' to 3' direction, just like DNA. So the side that's going to be copied is going to be the 3' to 5' side of the DNA double helix, because again, it's anti-parallel. RNA is ribonucleic acid. Sorry. Where DNA would be deoxyribonucleic acid. And the reason for that is it has ribose sugar, RNA does, instead of deoxyribose sugar, which DNA has. The 3 steps of transcription are called, if I can find where it says it on this slide over here, initiation, elongation, and termination, and that's what it meant by 3 steps on that other page you guys looked at. Polyadenylation was a vocab term on the other sheet. Again, just know what it means. And when we do transcription, if it's going to be a gene that's going to be transcribed, it's going to be the type of RNA that's going to be used as messenger RNA. If it's some sort of intron sequence that's going to be copied, it's going to be transfer or ribosomal RNA. It's not going to create a gene. It's not going to make a protein, and it just does a different sort of thing within your cells. And that will take us into my first little drawing here. So here was my double helix. So this was my promoter region. This is where this started. RNA polymerase, my little enzyme here, would have broken all those hydrogen bonds between the complementary nitrogen bases. I filled in some bases here. And then this open part, I think I missed in the notes the vocab term transcription bubble, but that's going to be a vocab term also. So just for practice, I'm going to go through and we're going to create our piece of RNA here, and it's going to be mRNA because we're going to make a protein. So just like we saw with DNA replication, we're going to have free nucleotides that are complementary being added to the existing strand. I'm doing them in red because the thing we're making is going to be RNA. Not that red is indicative of RNA, just so that it's a different color, you can tell that it's different. And so then as we fill in, it's going to be complementary base pairing the same way that it was when we did replication, just the big difference being that what we're making is RNA, so no thymine. So with guanine, it's going to pair with cytosine, guanine, that's a G, it's pretty hard, bad G. Thymine is going to be adenine, cytosine, guanine, adenine, uracil. So there's our big switch. You can still have adenine. If you have a thymine in the DNA, you're still going to have an adenine in the RNA. But if you have an adenine in the DNA, it's going to pair with a uracil when you're transcribing. So it's going to be a cytosine, adenine, guanine, again, uracil, adenine. And so then after this is complete, all those pieces are going to get connected together like what we saw with replication. Those hydrogen bonds in the middle are going to get broken. And then this little piece of mRNA is going to, and I drew it over here. So this is the piece of mRNA that we're going to have translated into a protein. And if you see these extra adenines at the end, I did my little polyadenylation at the end of my strand. And so again, this whole thing is in the nucleus. Everything, the DNA is in the nucleus. So this is in the nucleus. So our piece of RNA is going to find a nuclear pore, and it is off to find a ribosome, and we're going to get into our second step of gene expression called translation.